Hi, Wynand. Great to have you on today to discuss the field of microscopy in Africa and look at the different industries involved. Now, personally, I've been, last time I used the microscope properly was probably back in university for some kind of Giemsa stain on malarial parasites. Can you maybe give us an overview of the different types of industries that are using microscopes and, how they are impacted?
Yeah, thank you, Jeff. Thanks for having me on. Basically, yes, like you mentioned, mainly we focus at universities, but any industry that basically uses a lab will use a microscope. So here at Separations, we're basically focusing from life science, applied industry, and diagnostics. That's basically where we focus our trade on in.
Okay, no, awesome, excellent. And now, why do these companies use microscopes? Is it quality control aspects? Is it checking for microorganisms? Give us a bit of an overview on that.
No problem. So when it comes to, for instance, let's start with life science. There's a lot of cancer going on. COVID that happened. So life science, our customers will use it for doing some research. From live cells, cell culture, zebrafish research, from applied market where they would look at car manufacturing, for instance, for any defects on a car part, for instance. And even on diagnostic side, where you would go for a blood test, they will use a microscope. So this is basically the area that we cover here at Separations.
Okay, awesome, excellent. So now what I'm going to do is I'm going to obviously, I know we had a quick run through in the lab, so I'm going to share that information here and bring up that video.
So over here we actually have the Leica DMi8 with the Thunder application. It's a high-end fluorescence microscope where you can do computational clearing. Basically, computational clearing is all about, this is your conventional fluorescence image. With the computational clearing, it takes all out of the focus image, put it into focus. And as you can see, you can get far better quality on your fluorescence image. So this is all done via the software. This is an inverted microscope, and basically that's what the Thunder is all about. Okay, so over here we have the Leica DVM6 microscope. This is a combination of a stereo and a compound microscope. So as you can see, this microscope, we can use it with transmitted and reflected light. The nice thing about this microscope is you can tilt the head to get a better image. As you can see, you can get far better definition on your sample. This microscope can be used for any application, mainly in the applied market. With this microscope, you can even do a tile scan on your sample to give you a better overview of your sample. Here we have the Leica EZ4 educational stereo microscope. This microscope has a 5 megapixel camera built in. This is a Wi-Fi camera. It's got SD capabilities, Ethernet and USB. Also has transmitted and reflected light built in. Moving on to the S9. This microscope has fusion optics with a built-in 10 megapixel camera. You also have transmitted and reflected light on this stereo microscope. To our diagnostic microscopes, here we have the DMi8. This microscope is purely built for ergonomic use. To our Leica DM500 educational microscope, this microscope is purely used for educational purposes. Moving on to the Leica DM750p, this microscope is purely for polarization. Here we can use this microscope for geology purposes. So for any application, please reach out to Wynand at Separations, where we can help you and assist you with any Leica application for your microscope needs.
Cool. So Wynand, I know that there are different formats of microscopes, as we just saw in the demo lab. Can you maybe give us a bit of a breakdown of all those different applications?
100%. Let me share this with you, Jeff. So basically, when it comes to microscopy, there are basically 3 microscopes, different type of microscope we'll be looking at. So we have an upright microscope, we have an inverted microscope, and we have a stereo microscope. So just to show you basically the difference, how these type of microscopes look like. So as you can see, the compound microscope, this is where you would use a microscope slide. where you move on to a stereo microscope. That's where you would use a raw material for more, depth on a sample. And then where we move on to an inverted microscope is where you can use from slides to flasks to cell culture dishes. And these are the type of microscopes that we'll use in our industries. So just to explain to you the optical beam path of these type of microscopes, because it's all about the optical path and the type of samples you would use. So as you can see on your left-hand side, your right-hand side, it could be for your glass slides. So we have an option, you have light from the bottom, it goes right through your sample to your eyepieces, and that's the type of sample you would view. Where we move on to the inverted microscope, where you have a light source from the top, so your light will go through your sample, reflect back to your eyepieces. Okay, so these are the compound and the inverted microscope. Let's just move on to the stereo microscope. There are two different stereo microscopes, Jeff. One is called the Greeno and the one is CMO. It's the common main objective. As you can see, the CMO stereo microscope, it basically has an objective lens underneath the microscope. So you have two optical paths to the objective onto your sample. Where on the Greeno stereo microscopes, you have your two eyepieces, two optical paths, but no objectives onto your sample. So these are basically the two different or three different microscopes applications that we have.
Okay, awesome, excellent. Now I've heard about Leica in terms of, their microscopes, but I also heard about their photography, their lenses. They're synonymous for high quality lenses. What are some of the standout products? I mean, we had a nice look at some of them in the lab just a moment ago, but in terms of additional standout products, what really comes to light when you think of Leica microscopy?
Jeff, thank you. It's very exciting for me to share this with you. So in the past year, Leica actually launched a new confocal system. As you can see here, it's called the Mica system. It's a nice enclosed confocal system. And this is basically the second new confocal that Leica launched in the last 3 years. So this is quite exciting. As I showed you in our demo room, we also have the fantastic Thunder system. This is a very high-end fluorescence microscope where you can do computational clearing. And then also a market that we as Separations are very strong in is our forensics comparison microscopes. As you can see here, all you do is basically for ballistics application, you can compare two bullets, for instance, to each other here. And then something that I'm very passionate about is when it comes to IVF. So we have a system, we can do in vitro fertility as well. So these are basically the areas that we are really strong at.
Okay, awesome. Well, it was great you mentioned the one about the forensics. I mean, comparing ballistic, cuts on the as it leaves the barrel of the bullets. I mean, it's such an interesting field. I mean, for myself in the pharmaceutical world, I tend to hear a lot of like that inspection of raw materials, certifying and checking the grade. But the industry is impacted with microscopes. So for anyone who is interested in knowing a bit more, even coming through to our demo lab and actually seeing what your samples might look like under our different selections of Leica systems, please reach out to Wynand. I'll include his information in the links and the video description. And be sure to check out Separations channel, where we'll be highlighting the advancement of science in Africa. Thank you, Wynand.
100%. Thank you so much, Jeff.